In the second part of Chapter 11, we're going to talk some more about Mendel and his key discoveries, what we now often refer to as Mendel's Laws of Inheritance. And his first big insight that he had was an idea that we call segregation. Now, segregation is a word that also means separation, and this is an idea that makes complete sense if you understand meiosis. The incredible thing is that Mendel discovered this through experimentation before meiosis had even been discovered. The key concept in segregation is that during meiosis, when gametes are formed, only one gene per trait gets passed down into each gamete. So even though each person has two genes per trait, that you got one from each parent, this is how you get one from each parent, is by passing one gene to each gamete from the two that you have. But again, Mendel didn't know about gametes, and certainly was only beginning to understand what the idea of genes would even be at this point. What he did was, all through experimentation with pea plants, discovered this by doing something called a monohybrid cross. And at the same time that he discovered segregation, he also discovered the law of dominance, which describes what we've already discussed with the fact that some alleles are dominant and some are recessive. Now, what a monohybrid cross is when you take two individuals who are pure breeding individuals, like pea plants naturally are, and if they're each purebred for a certain trait, and then you cross two different ones with each other, the offspring are referred to as hybrids. A hybrid is the more formal term for basically a mutt, something who is the offspring of two different individuals. And a monohybrid refers to these two individuals being different for just one specific trait that we're looking at. Now, to keep everything straight, we need to kind of start to label our generations, or this gets very confusing. So the original generation, the parents, we can call them, are referred to as the P generation. Now, that letter P doesn't stand for a pea plant, and it doesn't stand directly for parent. It's actually a symbol that Mendel used that means father. It's actually pater, the pater generation. But we still just use the letter P as kind of a little nod to Mendel for his work, the symbols that he used. So whenever we start an experiment that is multiple generations, the first generation we call the P generation, and we take plants that have contrasting forms for a trait. So maybe one plant is tall and one is short, and if they're both purebred, then we want to see what the offspring will be like. And if they are both purebred, we find out that one form of the trait disappears in that next generation. The next generation is referred to as the F1 generation, and it basically means the first generation of brothers. Now, if it seems weird to hear them talking about father and brothers, keep in mind Mendel was a monk, and he was working among men all the time. It probably was pretty normal terminology for him to use. But now, Mendel couldn't understand right away why this one form of the trait would disappear. For instance, if you cross the tall plant and the short plant, you get all tall offspring. Well, what he eventually figured out, and that makes sense to us now, is that all of the F1 plants are heterozygous. They each have one dominant allele and one recessive allele. And the reason for that is because in the P generation, one parent had two dominant alleles, the other one had two recessive alleles. The only way that they could pass on their offspring is for one of those parents to take the only allele they had, which would be in this case a big A, and the other parent to pass down a little a, and that's the only possible combination you can get for those offspring. Now, when you get to the next generation, it gets a little more complex. The F2 generation you get by taking two F1 plants, which again are both heterozygous, and crossing them with each other. When we do that, you start to see both forms of the trait showing up again. You see some tall ones and some short ones. And another key breakthrough Mendel had was he started counting those plants and realized there was a mathematical ratio here. The ratio he found was a 3-to-1 ratio. 3 or 75% of the offspring showed the tall trait, which was dominant, the one showing up in the F1 generation, and 1 or 25% of the offspring would be showing the recessive form. Now, the key thing this did in Mendel's time period is it proved for a fact that blending doesn't occur, because not only when he crossed the tall and the short plant did he not get medium-sized plants, but both the tall and short hereditary information were being maintained in the F1 generation, because in the F2, when all the parents were tall, we see some short offspring showing up. So that means that recessive allele has to still be in the F1 generation, even though they aren't showing it. The term we often use is we say they aren't expressing the recessive allele, they're only expressing the dominant allele in the F1 generation. Now the reason you get that 3-to-1 ratio and that so precise mathematical ratio is the fact that every sperm has an equal chance of fertilizing any egg, and when the sperm and egg are formed, there are equal chances of each different allele showing up in there. Since the F1 plants had a dominant allele and a recessive allele, and they pass exactly one on, each sperm had a 50-50 chance of dominant or recessive allele. Each egg has a 50-50 chance of dominant or recessive allele. And if you're good at math, you can start doing these in your head. If you're not good at math, it's helpful to have a tool. And that's where something called a Punnett square comes in. If you remember Punnett squares well, you can probably skim past this next little section pretty quickly. I'm actually going to follow a link that talks about Punnett squares that I found this on a website from PBS. This is at pbslearningmedia.org. If you Google it, I'm sure you can come across it pretty easily. And it has some information about Punnett himself. And Punnett, who's pictured here on the left, is going to kind of go through in his words explaining this tool that he's using. And I'm just going to skim through the text here. It's basically explaining a lot of what we discussed already. But the tool of the Punnett square is to describe how if you have two individuals that are heterozygous, here instead of tall and short, he's using yellow peas that are each heterozygous, and how we can use his Punnett square tool to work this out. Now, I've never seen a Punnett square drawn quite like a tic-tac-toe grid before. Usually we would see lines connected on the right side and the bottom here to truly make a square. But what we use is the grid to kind of lay out each possibility. And the possibilities for the first plant here to give either a dominant or recessive, and the other plant to give a dominant or recessive. And then in the other boxes, we fill in the combinations that could show up. Now, remembering that the big Y is dominant, we can see 3 dominant combinations in terms of the phenotype to only 1 recessive. And they're going to try to make this even more clear for you here by kind of going through and highlighting everything and then also putting it in kind of picture format. Well, I think we can kind of skip back through. I think we can all picture what the different combinations of alleles would be for each of these. But if you are having trouble with Punnett squares, I would highly recommend going slowly through and reading through this website from pbslearningmedia.org. And I'll try to remember to put a link up for that on our website so you can find that even easier. But back to our notes. Another key idea that Mendel had is something that he referred to as a test cross. And the idea for a test cross is, more than anything else, actually a way to kind of help you keep track of what organisms you're working with when you're doing genetics experiments. Now here's the issue. If you have an individual who shows a recessive phenotype, a short pea plant, or a pea plant that produces green peas, because green peas are actually recessive, we know the genotype there. The genotype is 2 recessive alleles. We can just use a generic form here to say little a. If you show the recessive form, it's easy. The problem is, what if you have someone that shows the dominant form? If an individual shows the dominant phenotype, we don't know whether they're homozygous dominant or if they're heterozygous. It could be either one, and there's no way to tell, short of breaking down their DNA and analyzing the specific genes. And to be honest, the technology isn't really quite there for that at this point, even today in 2013. But definitely not back in Mendel's time. So he needed to experiment. The way he did this was he took a recessive individual and crossed it with his unknown individual, and then using the whole set of combinations that could show up, you could determine what that unknown genotype was. Let's take a look at that using a Punnett square. So let's say Mendel has a tall pea plant and he wants to find out its genotype. So he's going to cross it with a short one because, again, we know the short plant has a little t genotype. We don't know what the genotype of the tall plant is. We know there's a tall allele, but the second allele could be tall or it could be short. So let's make our Punnett square. and we'll fill in the possible alleles that each parent could pass on. So for the short plant, it's always going to be a little t. For the tall plant, it could be a big T, or it could be whatever that unknown one is. And at this point, we still don't know, so let's fill in the Punnett square. In the first two boxes, we can see those offspring will be heterozygous and they'll be tall. Very straightforward. The other two boxes, it all depends on what that unknown allele is. But if you actually perform an experiment between these two pea plants, we can figure out what the unknown is. If any of the offspring show up short, we know that that question mark allele is a recessive allele. We know that the tall plant was heterozygous. If all of the offspring end up tall, we could maybe assume that that allele is a dominant allele. It really depends on how many offspring. If there are 5 offspring or 7 or 10, we can be a little bit certain, but it's really tough to say. If there are only 1 or 2 offspring, you definitely can't tell this way. But if you have hundreds of offspring and every single one is showing up tall, then you can be quite certain that that unknown allele is a tall one. Now, we don't do test crosses with humans. For instance, in eye color with humans, blue eyes are recessive. But if you have brown eyes, it's really not a good idea to find someone with blue eyes and breed with them and have a bunch of children just to find out what your unknown genotype is. We have other ways of finding that out other than doing test crosses in humans. So number one, it's kind of unethical to have kids just for the sake of finding out what unknown genes are. But secondly, humans don't really have enough offspring to get really reliable results unless you see that recessive form show up almost immediately. Okay, back to Mendel. His final really big idea he came up with is the idea that we call the law of independent assortment. And this is now recognizing the fact that organisms don't have just one trait. You have multiple traits. And every trait has to go through the processes of inheritance just like every other one. So Mendel experimented in looking at 2 traits at the same time. And instead of doing a monohybrid cross for these experiments, he would do a dihybrid cross, where you're looking at 2 traits and you're doing a hybrid cross for each one of those traits simultaneously. So he predicted that the F1 plants, when he did these experiments, would show dominance for both of those traits. The reason is each trait is kind of doing its own thing. If you have, let's call it trait A, and one parent is homozygous dominant, the other parent is homozygous recessive, we know that offspring is going to be heterozygous for trait A. Same thing's true for trait B. And he predicted correctly that's what we would see. But in the F2 offspring, things get much more complex. You actually start seeing a ratio that we describe as a 9 to 3 to 3 to 1 ratio. And that math looks messy, so we probably want a tool to help us out. A Punnett square is definitely going to come in handy with this. So let's take a real example. We use pea plants again. And in the pea generation, we're going to have the first parent be dominant for both traits. We use round peas that are yellow, and round is dominant, so we use the letter R. Yellow is dominant, we use the letter Y. We're going to cross that dominant homozygous round yellow pea plant with a plant that has wrinkled peas that are green, and those are both recessive traits. Now, in the F1 generation, all of the F1s, like we already said, would be heterozygous for both traits. There's no other possibility that can show up. And in terms of phenotype, that means that these F1 plants will all look like the parent that was dominant for each one with round and yellow peas. When the F2s, if we cross 2 F1s with each other, that's where things get a little more complex. So we need a big Punnett square. Here in small type, we have what we just said is going to be our experiment. And we have listed here the 4 different possible phenotypes we can get. We could get round yellow seeds, round green seeds, wrinkled yellow seeds, or wrinkled green seeds. And in doing our Punnett square, we're going to take each parent and look at every possible combination they can pass down. Remember, law of segregation says you pass down 1 allele per trait. So each parent has to always pass down 1 R and 1 Y. You can never pass down 2 R's or 2 Y's. It's always 1 allele per trait. So in math class, if you learned about foiling, we can use that same idea here. We can foil the letters we pass down. We can do the first ones, the big R and the big Y shows up over here. The outside ones, big R, little y. The inside would be little r, big Y. And the last one's little r, little y. And because both parents are the same in this experiment, the other parent's going to give us the same combinations. Now, this gives us a pretty complex square here, and if you notice in our Punnett square, it's not 2 rows by 2 columns, it's 4 rows by 4 columns. Every time you add one more trait, it doubles both the width and the height of the Punnett square. So if we try to do 3 traits, you'd have 8 boxes by 8 boxes. 4 traits would be 16 boxes by 16 boxes. Those get really messy, really difficult quickly. We won't do any of those in this class. And anyone who actually does experiments on those is definitely going to use computers to help them out. But if we look at this, when we come up with all the combinations, I've highlighted in yellow the round yellow seeds that show up, and if we count them, we see there are 9 different combinations that can show up of round yellow seeds. There are 3 combinations of round green seeds. There are 3 combinations of wrinkled yellow seeds, and only 1 combination that will give you wrinkled green seeds. So in other words, that 9 to 3 to 3 to 1 ratio is describing the 4 different phenotype combinations that show up. Now, it looks complicated, but remember, all we're saying is we're looking at 2 traits at the same time, but each trait is still doing its own thing. What if we just focus on the shape of the peas, whether they're round or wrinkled? If we just focus on those, we've got 12 different round pea combinations showing up, highlighted in yellow, and 4 wrinkled ones. Well, if you take the ratio of 12 to 4 and simplify it, that's a 3 to 1 ratio. So in other words, the shape of the peas is still doing the same combination it was before when we looked at just 1 trait. But when we describe 2 traits simultaneously, that's where we get this 9 to 3 to 3 to 1 ratio. If we would actually go back and do the same thing for color, we'd see also 12 yellows to 4 greens. We're still seeing a 3 to 1 ratio for each one. It's only when we combine them we start seeing more combinations. Now keep in mind, humans have probably thousands of genetic traits. We don't know the exact number, but maybe the ballpark of 20,000 or so. That's going to explain how you get so many different combinations that can show up to make one human compared to a brother or sister so different based on all those different combinations that can show up. Now the reason we call this the law of independent assortment is what we're saying is each gene is doing its own thing. The shape of the pea was being inherited on its own, and the color of the pea was being inherited on its own. It's only when we try to look at them simultaneously that we see all these different combinations, but each one still had a 3 to 1 ratio. Now, Mendel thought this was always true. We know today, as we understand chromosomes, this isn't always true. The only time this is true is if those genes are not on the same chromosome. So if one is on chromosome number 1 and another trait's on chromosome number 7, they're going to be assorted completely independently. But if they're both on chromosome number 1, sometimes they're going to travel together. So if they're on the same chromosome, they're going to be what we call linked together. And we're going to talk more about that when we get to our next section here in terms of some differences that come up that make it look like independent assortment is not taking place. But it's only the fact that these things are physically connected sometimes that means that they do sometimes have to be inherited together. Well, that's the end of our part 2, a bit of a long section there. But those are all of Mendel's real key ideas that lead to us calling him the father of modern genetics. But in part 3, we're going to take a look at some things that Mendel missed out on because they didn't show up in the experiments he did. As always, if you have questions, bring them to class, and have a great day.